Thank you, everybody, for joining us this afternoon. The Graduate Student Division is happy to welcome Dr. James Harper for this month's Pioneers in Dairy Science webinar. Dr. Harper is Professor Emeritus at Ohio State University. He's done a tremendous amount of work over the past 70 years, which he's going to share with us. So if you're ready, Dr. Harper, go ahead and take it away.
Okay, thank you. Welcome, everybody. It's a pleasure to be with you today to go over my 70 years working in dairy foods. I began working as a lab technician at Purdue University in 1942. And I was very privileged to grow up professionally during a period of instrumental method development and had the opportunity to use many of these over the years. I'm going to talk about the work from the very beginning, when in 1941 I began to work as an undergraduate with Professor Eppel at Purdue University. And the work is covered in many areas, but generally involving investigation of industrial problems and always alert to new technology. Our research is no better than the methods we have available, and many of these have been developing over this period and are still new methods developing. So I've worked with milk and cheese and ice cream, worked with proteins, worked with dairy waste and flavor, and in a sense, the work has divided into sections over time, frequently related to the problems that were available or actually in progress at the time, or involving opportunities for research. You always have to worry about funding the research, and it was funded by many different areas. There's been effort to work in different areas, and all of this actually has related to either milk, cheese, ice cream, milk proteins, dairy waste, and or flavor. What I'm going to do is to talk about the decades of research and indicating the methods that were evolving at that period of time and what my research involved in. Actually, as a student, had the opportunity to develop a method for determining higher molecular fatty acids. At that period of time, the farmers put their cans of milk out on the road, and they were picked up and taken to dairy. FDA actually then developed a very classical, difficult method, and so we worked on a new one. I see I've jumped the thing forward, and I'm not clever enough to get it back, but we've jumped into my work at the University of Wisconsin, and I'm not going to read all this. You can read it at your leisure. But basically, When I started my graduate school in Wisconsin, I was given the opportunity to work on cheese. At that time, we were interested in amino acids and their role in cheese, and long before we understood the fact that amino acids have more than one role. So there was a method developed in Wisconsin called microbiological assay. And this meant selecting lactic acid bacteria that had the ability or the need for an essential amino acid. And then you could actually use different concentrations, develop a standard curve, and apply that then to a cheese extract. So, in the 1950 to 1960 period, we're seeing the development of gas chromatography. We saw radioisotopes become available as tracers and begin to see application of mass spectroscopy. I joined Ohio State University directly out of graduate school in Wisconsin in 1949. At that time, the Italian cheese industry was having problems because the FDA had embargoed a material called rennet paste. And rennet paste is the ground up contents of the stomach and was used in making Romano and provolone cheese. When the FDA did this, confiscation, the cheese industry could no longer make the flavor in their cheese. And so actually, when I graduated, I had the opportunity then to go to Ohio State as an assistant professor, but most of the funds actually came from a contract work with the U.S. Department of Agriculture. And so I was an agent initially. And when I went to Wisconsin and visited the cheese factories, I found that the flavor in the cheeses they still had used rennet paste tasted like fatty acids. So that there were no methods available at the time. So we developed a column chromatography method. And then in the same decade, we also started to use radioactive tracers because they had high sensitivity. Tried many different chromatographic methods, and before the end of the decade, had also built our first gas chromatograph. The silica gel method was used for many. This is here for your later perusal, if you wish, but by using the fatty acid method, we were able to actually determine that the lipases from kid, calf, and lamb had a selectivity for lower fatty acids. And we show butyric acid here as percent of the total. Butyric acid in milk fat's about 10% on a molar basis, but you get up to 30-40% by using the lipases from the stomach. There was a gland at the base of the tongue that actually excreted lipase to the ruminant young. And this is what made the Italian cheese or Romano provolone have the particular flavor that they obtained. So we could see a selectivity and see differences in the lower molecular fatty acids. By using this methodology, we were able to actually get the Italian cheese business back in operation. At about the same time, became interested in the availability of radioisotopes because of its high sensitivity. And one of the first uses was actually to work with a colleague named Seiberling, who was interested in trying to automate the dairy plant. And he decided that if he could find out how to clean a automated valve, that he could actually use that then to install automation into the dairy industry. And also looked at other chromatographic methods. So it was a busy time. It was a fun time. Looked at various other methods related to cheese. Looked at amino acids by column chromatography. You've never seen this, I don't think. We didn't have much money to work with, and so we figured out how to make our own gas chromatograph using a column, using nitrogen pressure, and actually pushing the solvent through the column, so we got selective removal of individual volatile fatty acids, and use an automatic titrimeter as our detector. It was only good for fatty acids and amines, but until we could get solvent enough to buy a gas chromatograph, this is the way we okay. So our isotope work spread out from P32 and calcium to carbon-14, sulfur-35, and tritium on occasion. Also, in the beginning, we made our own uniformly labeled amino acids from bean sprouts and isolated compounds by ion exchange chromatography. Moving on to 1960, 1970, GC methods coming in common use, high pressure liquid chromatography introduced, infrared spectroscopy being used. And so we used radioisotopes in other research. And going back, we used calcium-45 to prove the efficacy of CIP. We tried to use P32, but it reacted irreversibly with the stainless steel. Then we used paper chromatography, thin layer, other methods for cheese, culture products. Throughout the years, the work has somehow always come back to cheese. The other members of the department had developed a cheese slurry system to accelerate cheese ripening. And we worked with this to find out the mechanisms and found out that actually 1 day of the slurry at 30 degrees centigrade was equivalent to about a month of ripening. And then also explored infrared application. So we're talking about what happened over a 10-year period. And this is Bessie. This is me. This is another colleague at Experiment Station in Worcester. And so we used carbon-14 and used sulfur-35 to either eat or inject into the bloodstream of the cow to get radioactive milk. And then I would transport this from Worcester, the 90 miles back to Columbus. Much of what we did with isotopes in those days, which was under approval of the agencies governing use of isotopes, would not probably be used today. Use sulfur-32 to label proteins, and that's what we're seeing in the picture. Was used to induce for the effect of heat. We looked at culture products and product degradation in cheese. And also at different times used P32 and C14. This merely indicates the kind of compounds that were produced, and using different sources of sulfur, either salts, amino acids, to get different labeling patterns. In looking at the sulfur compounds, and we could separate the sulfur compounds into different classes by putting them through different traps, so that each trap would collect a different classification of compounds. And at the end, we actually looked at carbonyl compounds. And looking at culture activity, that we could find out what the compound precursor was converted to by the starter organisms, so the glycosylate became acetoacetic and acetate, and pyruvic acid and oxaloacetate, and so forth. And then we could show that different starter organisms could produce different compounds. They all produce glutamic acid, and they all produce leucine. But the others varied according to the starter use. We begin to see some selectivity. And this is, again, some additional data. You will have in the webinar material a listing of the publications, including the ones that are represented here. Move on to 1970 to 1980. Again, continuing the development of new methods. And at that time, funds for research in dairy flavor and other related dairy were very rapidly disappearing. In 1972, the Clean Water Act began to focus for the dairy industry a real problem with whey. The biological oxygen demand for human waste is about 200 parts per million. The oxygen demand for whey is 36,000. So that you would have a single dairy plant in a fairly large city producing more BOD waste than the entire other parts of the city. So that we work with the Environmental Protection Agency in this area. So research moved away from flavor, but still went on with some cheese slurries and isotopes, and began to have the opportunity to get a real gas chromatograph and could begin to look at the laundry list. And there were hundreds of compounds that were being produced in cultures and in cheese that were being investigated by gas chromatography. This is really a laundry list that you can look at a later point of all the compounds, and until only recently, the actual role of these compounds in the flavor could not be really determined very accurately. By 1980, most of our research was in the area at Ohio State in waste treatment. And I had been working with cheese whey and ultrafiltration, and happened to have the opportunity to get funding for a whey workshop. It brought in people from Ireland, Canada, New Zealand, and Australia in 1975. One night, it took me 2 hours to get home. It was a snowstorm, and I got in. I recognized the voice on the phone. It said, "Jim, what would it take to get you to New Zealand for a year?" I told him tonight it wouldn't take much. So I went to New Zealand, spent 5 years in New Zealand, and working with whey protein development and how to manage ultrafiltration to give different functionalities. I lived in New Zealand for 5 years and then continued working with New Zealand as a consultant for the next 5 years. During the recession in the early 1990s, Ohio State University did not replace anybody. In 1992, I was asked by the chair of the department to teach some of the required courses, and which I did. And then was invited to take an endowed chair in dairy foods, which I held for the next 20 years. So 1990 to 2000, electronic nose appeared in 1994. Solid phase microextraction came into use. Improved selective GC detectors, chemical basis of flavor began to be understood, and Fourier transfer infrared was in common use. I rejoined, I stayed in 1992, became involved in electronic nose research, and we could differentiate using electronic nose from different factories, different ages, different flavors, but had no method by which to determine what the compounds really were. At the same time, the Swiss Cheese Consortium that had been started by a PhD from Utah that was working with the largest Swiss cheese company in the world here in Ohio, moved the consortium to Ohio State. And so for the next 20 years, one of our focuses was always Swiss cheese. Between 2000 and 2010, analytical methods were continuing to develop. Sensory descriptive analysis, coupled with GCMS and model food systems, were showing promise. FTIR, from my research, electron nose actually shifted from those available to using GC mass spec and to find the masses associated with differences in milk and cheese flavor. And the cheese research continued with the Swiss Cheese Consortium. Polyfunctional thiols were found in cheddar cheese using a special sulfur detector. A selected ion mass spectroscopy was introduced in 2008. We were asked to evaluate this. And at the same time, we were also using FTIR. So during this period, we found that we could actually combine FTIR for water-soluble compounds and SIFT-MS for volatile compounds to begin to get a better understanding of the factors that differentiated the volatile compounds, and eventually begin to associate that with cheese flavor. I won't dwell on these. This is just the fact that we could associate different masses with different compounds. Sulfur compounds frequently in parts per billion and parts per trillion. Concentrations are above the threshold of flavor analysis. So we looked at cheddar cheese and found about 40 new thiol compounds in aged cheddar cheese. And 20 of these were tentatively identified. We still have yet other compounds that we need to identify. So that each compound has a different descriptor associated with it. So we're still looking at now using the SIFT mass spec, trying to determine the role of these different compounds in cheese flavor. During this time, the Swiss Cheese Consortium haven't been moved, still currently in operation. We have 8 members in 6 different states. Work done in Dr. Rodriguez's laboratory to show that for commercial cheeses we can actually differentiate them on the basis of strain of the culture, the basis of factory age, and the basis of flavor. The results of this work really is water-soluble, non-volatile compounds, and we couple that with the SIFT-MS to get the volatile compounds so we get a more complete picture. Methods are still improving. We begin to get a better understanding of the chemical basis of flavor and continue new approaches to use SIFT-MS. Note that I first spelled it with a C, but it really should be an S. Odor activity value, which is the concentration divided by the threshold, becomes an important concept because we found that for compounds with an odor activity value greater than 1, that is, the concentration is greater than the threshold, we have a high impact flavor compounds. And one of my former students is just putting together a paper showing the relationship between this and sensory of cheese from 5 different factories. One application of SIFT-MS is relatively new. We can go down to parts per trillion in real time without any sample preparation. And so this is becoming very important tool in both flavor and in understanding what the starter cultures do in cheese. So basically, this is an outline of the methodology. So that we start with a plasma with different ions that we go through a quadrupole to sort them out, and then go through a second quadrupole, and can actually use mathematically the mass spec then can actually determine the concentration of the various compounds. This is just additional information on odor activity, which I believe has a good future in flavor research. And the volatile compounds in aged cheese are very variable and very high. They're diverse, and they'll be different in different factors and different ratios, which makes for different flavors. This is a rather complex graph that really actually shows 5 different cheeses separated from each other, and this is the compounds that cause the differentiation. I've gone through a lot of material very briefly, and happy to answer any questions that you might have. And can't mention my career in academia without mentioning students. Graduate students and undergraduate students have been an integral part of my research. Much of the credit, if not all of the credit, for the accomplishments of the research and publications really belong to the efforts of the students. I've had over 150 MS and PhD students, and currently, as Professor Emeritus, much of the research that I'm continuing to do is being conducted by juniors and seniors in the department. There is a list of publications that lists over 300 different journal articles, and that you can take a look at if you want to more detail of what we've been covering. I greatly appreciate the opportunity to involve in this webinar. I have to admit, this is the first time I've been involved in this type of operation. When I sat in your place, we didn't have even electric typewriters. The computer was primarily a thing of the future, as it was TV and other things we take for granted, such as the cell phone I imagine that all of you carry. So now let me open it to questions.
All right. Thank you, Dr. Harper. I appreciate your taking the time to talk to us, and I guess I also have a little personal stake in thanking you because I'm sure you've had a hand in developing a lot of the techniques that I use for my research into off flavors and dairy foods. So while we wait, there's just to remind everybody that if you go to the top of your screen, there's a drop-down box that has Q&A button on there. You can type in your questions there, and then I'll get those to read those to Dr. Harper. Yep.
Kevin, can I also get a copy of those electronically sometime so that I could perhaps get more information back to the people that are interested?
Yeah, we can do that for you.
Okay. Well, first off, I guess I have a question, kind of for selfish reasons, I guess. Since we have such a expert in the field of flavor analysis, and I mentioned I do research in the area of off flavors in dairy foods, primarily in milk, but in your experience, I guess what do you think the most reliable methods currently available are for identifying off flavors and something like milk? We've used both the trained sensory panel, and then I've dabbled a little bit in doing solid phase microextraction work, but have kind of found the solid phase microextraction to be really variable and hard to interpret.
Yes.
So do you have an opinion on what you think the best method available is right now?
My goal from the beginning was to try to understand the chemistry of flavor, whether it be cheese, milk, or whatever. And we made some progress in that direction, but we still have a long way to go. We begin to believe that, I would say, any method that you can use to quantitate that can be converted then into odor activity values gets us closer to an understanding of chemistry. I'm sure you know Marianne Drake, and she has worked with us on many of our cheese with her sensory panels, and we begin to believe that there's frequently more than one compound contributing to a particular sensor response, which means we may never be able to get the complete chemistry. But we have found that odor activity values are being useful in being able to differentiate between the results of flavor panel and the cheese flavor on a sensory basis. I don't know if that helps you or not.
No, that's kind of what I've I guess figured or had thought had been the case, but I thought, well, I had the opportunity to ask an expert, I would just check. This odor activity is interesting, and I'm going to look more into that after this.
It's a difficult thing in that you get different threshold values depending upon the method that's been used to determine them. And so what we've turned to is basically trying to find 3 or more threshold values that agree with each other within one order of magnitude in making these determinations.
Okay. Looks like we have a question from Doug Snyder. He says, thank you for providing the presentation. Since elevated levels of copper in the diet cause an oxidized flavor in milk, will that cause a detectable off flavor to cheese or yogurt products as well?
In cheddar cheese, it only takes above 4 parts per million to get an off flavor. But Swiss cheese, particularly when it was made in copper kettles, would you go up to several hundred parts per million copper and not get an off flavor. So it depends upon the cheese. When these people are asking questions, it would be helpful to me to know where they're from. My contacts around the world and around the country are mostly gone, either retired or totally gone. And so many of the new people I really don't know, and I would like to.
Doug Snyder is from the university I'm at, Iowa State University, Vet Diagnostic Lab.
Okay, thank you.
Mm-hmm. All right. Do you have any other questions for Dr. Harper?
My email is harper.9@osu.edu. I would be happy if we went through a lot of material very quickly, and I'd be happy to try to answer any questions you have, even after the seminar is long over.
All right. Well, I'm not seeing any more questions, so I guess I'd just like to say thank you again for all the work you've done. It's definitely had an impact on my research program. And
thank you. It is difficult to put it together.
Yeah. Well, since I'm not seeing any other questions, I guess we can conclude for this month's webinar. And thank you again to Dr. Harper for sharing his work